Good afternoon, everyone. Today I will give you, well, I'm Andrea Dalle Vedove. Today I will give you my presentation on some of the work I performed in the past. That is, and I will speak for about half an hour on a topic that goes from protein crystallizations to the structural characterization. As you see from the outline of the presentation, the title is a bit misleading since I will spill out a little bit from the title topic and also describe a little example of what can be done with structural biology for drug discovery. Before starting, since this is the first time I present some work for AREA from my new work position, a little bit of background about me. I got my degree in chemistry at the University of Padova, pure chemistry, doing internship in chemical synthesis. Then I moved to Milano and did a PhD in structural biology, protein crystallography. It was kind of a serendipitous switch, but a happy one, let's say, because then I kept my interest on that, and also did a postdoc in the University of Trento, where I kind of put together my past formation and enter in the group of structure-based drug design. And I will speak a little bit of the work I did in Trento in the last part of this presentation. Now, coming to the reason why I'm here on this stage, I work for AREA, but I work in the structural biology lab at the Elettrosynchrotron Trieste, where I collaborate with the two groups that are based in the synchrotron, that is the group of Paola Storci and Silvio Nesti. As you see, they work on very different topic and targets, but the work they do is characterized by the fact that they intensively use structural biology, and I'm there to help the different project with the protein crystallization, X-ray crystal diffraction experiment, structural resolution of the structures, and on with the analysis and extract the information that we gather from the structures. We can now start with the actual presentation. I always like to start with this slide, that is the experimental pipeline that we need to do when we want to do any kind of structural biology experiment. As you see, even from this very schematic depiction, is a quite long process to get to the pure protein and then to have crystals. You can have problems in more or less every step. So the work is kind of complicated. In this presentation, as you might have inferred from the title, I will mainly focus on what is on the bottom part of the slides, so the part from crystallization and on, and show you how to get crystals and which kind of information can be then extracted from the structure we obtain. So before starting with the experimental part, let's say, I just want to agree with you on what it is a crystal, that is a solid, where we have the constituents, the protein, in the case of a protein crystal, that are arranged in an ordered way, and this ordered unit is repeated throughout the whole crystal, so the whole solid. To obtain, experimentally speaking, a protein crystal is, from the technical point of view, relatively simple. The setup is not particularly fancy. We have multi-well plates, doesn't matter the number of wells, that has, of course, a well in which we put a reservoir solution that is a mix of an extremely different array of, let's call them ingredients. We may have buffers, salts, polymers, solvents, more fancy molecules. There is more or less no limit. And there is always, or a lodging, as you can see in the slide, the sitting drop bridge, or we can prepare the drop on top of the slide, you see up here sealing the well. The protein drop is constituted by protein solution, of course, and reservoir solution. The standard ratio is 1:1, but it is one of the variables that we have in the crystallization experiment. As I said, we seal everything. Now this is a closed environment, and through vapor diffusion, we have the progressive, let's say, dehydration of the protein drop. And so we have, as you see here on the right in this state diagram, we have a progressive concentration of all the species that are present in the drop, so protein and the different precipitant salts, and the system shuttles in the phase diagram until we reach first the supersaturated states, and then we can hopefully reach a condition where we can have nucleation events. And these are the first, very first extremely tiny crystal that then over time grows and hopefully become a fully developed crystal that we can use in our experiment of X-ray crystal diffraction. The component of the reservoir solution determines how the system moves in the phase diagram. If we have an excessive, let's say, dehydration of the drop, or the system, because of the temperature of the experiment is wrong, goes too fast in concentrating, we just jump the metastable area, the supersaturated states, and we have precipitation and we have nothing. If the conditions are, let's say, too diluted, we might never join the blue-green area here where we can have a nucleation event, and so we will never have crystals since we do not reach that state. Of course, as I said, it is since the variation of the reservoir solution is more or less infinite, we will have to try different conditions to find the right one for our protein to crystallize. And at least up to now, there are no tools that predict which mix to use for a given protein. So we have to try. There are, of course, commercial screens that more or less cover different chemical spaces, but trial and error is still the base for protein crystallization. That's why it may take like forever. Of course, we may also take a protocol, try to reproduce it, and the crystal that we end up in our tray are not the same as those in the publication. Nothing is completely lost, or at least not always. There are, of course, techniques to switch from ugly and unusable crystals, because for single crystal diffraction experiments, we need crystals that are big enough and of quality high enough to yield a good data set. But we can, of course, optimize. We have different possibilities. The easiest one are tweaking with crystallization condition or doing additive screen. Well, optimized crystallization condition means simply do slight variation of, let's call them ingredients of the crystallization mix, and see if these small changes yield some kind of better results from the starting one. We can do additive screen. Also, in this case, additive have the most wide meaning we can imagine, going from trivalent cations to extremely fancy metal complexes. The sky is the limit here. We can use what are called seeding technique. You might have heard micro seeding, macro seeding, strict seeding. They are all slight variation of the quite schematic and oversimplified scheme I did here. And it just means using extremely small crystal or even smash the crystals of bad quality, putting them in a new drop that I just set up and use them as a nuclei for crystal growth. In this way, I don't have to reach the azure green area. That means I can have nucleation, but I can have the nuclei I put in from the exterior start growing before that happens. And this can yield, as you see in this real life example of the last experiment we did in the lab, to extremely big increase in crystal quality. What you see is the same protein without seeding it crystallizing in those needle balls that are absolutely useless from the crystallographic point of view, but doing seeding, you end up having those huge crystals that are of an amazing quality. This is a very happy, let's say, experiment. Not always you end up in this situation, but it is a really good indicator on the power of this relatively simple technique. Or what you can do is use a nucleation adjuvant. Also, in this case, the span of different things you can use to help nucleation is very big, and also it can go from extremely fancy complexes to fibers that you just put in the drop. And here you can see crystal growing directly from a small piece of hair. The cylinder you see is a piece of hair, and crystals are using the roughness of the hair as nucleation point. As you will see, I will speak in, well, sorry, if nothing of this happens, you can use this approach I highlighted in red, so surface entropy reduction, optimization of crystal contacts, a redesign of constructs. As you can infer, at least from the last one, all of these approaches imply that you have to do, in some extent, mutation on the protein. And this means you have to start from square one, basically, and also redo all the purification and production process, sometimes re-optimizing everything. So they are available, of course, but you always hope not to be in the condition of having to use them. As you will see, I will speak about complexes with small molecule in the second part, but you can do complexes also with other proteins or DNA, RNA, whatever. Also here we have kind of different techniques we can use. The most straightforward you can imagine is co-crystallization, and it works exactly like the crystallization of the protein alone, with the only difference that we add in the drop also the ligand we want, be it small molecule, protein, DNA. We can actually or add it in the drop or form the complex before during, let's say, purification, and then use the complex solution as it is the protein solution. But the rest of the experimental part that is behind it is exactly the same. It works, it's straightforward. The only problem is that in some cases, it is like working with a completely new protein. So your optimized condition won't work, and you have to start back with all the screening I briefly explained before. When you are dealing with a small molecule or small substrates, also small peptides, you can go for a soaking technique. Of course, you have to have a protein that produces big enough crystals, but you can exploit the fact that the protein crystals contain a very big amount of water, of solvent. We can reach extreme cases where we have 80–90% of solvent in the crystals. The solvent mainly reside in channels that are formed in the crystal by the packing of the protein molecules. And so what you can do is just take your crystal, move it in a solution with a ligand, or add your wannabe ligand in the crystal solution. And then what happens is that by diffusion, your ligand reach the protein, form the complex, and hopefully you are able to use the crystal to collect the structure of the complex. One slight variation of this is what can be called back soak or back soaking. The only difference is that we start already with a co-crystal of protein plus ligand. We move it in a solution with a new ligand that I labeled the ligand B here for the sake of extreme simplification. What should happen, hopefully, is that by the combination of higher affinity of the new ligand and higher concentration in the drop of your new ligand, you have always by diffusion the switch of the two, and so you might be able to collect again the structure of your protein with these new ligands. There are a lot of caveats that I just not said yet, because for soaking and back soak to work, you also need to have packing compatibility with the ligand you want to analyze, because it may happen that your ligand is too big for the crystal packing, and so instead of having the crystal of a complex, you have nothing, because your ligand will just make the crystal explode, and so you have to change approach, of course. So all this work to be able to have this nice single crystal big enough to do the X-ray diffraction experiment, and then we go on with at this point computational pipeline where we do structure solution and start analyzing the crystallographic structure, and so we start extracting information from the structure we obtained, be it understanding how the mutation we put in that we took from a pathology impacting humans, animals, whatever, be it seeing how the ligand we co-crystallize is interacting with my molecule. I will analyze the structure, extract this kind of data, and then move on with my project. So we reach the second part of my presentation, where I will show you a slice of what it is the process that is used in structure-based drug design. I will do it showing a little bit of work I did in my previous position on the development of inhibitors for BAS2A. And so before starting showing what we did, I will just focus a little bit what we are speaking of extremely quickly and most likely oversimplified. But so BAS2A is a bromodomain that is a class, a relatively big family actually of epigenetic readers that reads acetylated lysine in histons, and by doing this and of course interacting with the rest of protein machinery, they regulate transcription and chromatin remodeling. With BAS2A, but it was also seen with other bromodomains such as BRD4 or BET. Some kind of cancer for BAS2A, prostate cancer, have an overexpression of this bromodomain that's cascading. It induces an aberrant gene silencing that is an extremely bad prognosis marker, as you see, because it is correlated with aggressiveness, development of metastatic phenotype with the recurrence of the tumor. And this is the reason why we are investigating. Well, now they are investigating since I moved from that place. BAS2A inhibition is that knocking down this protein impairs somehow the metastatic potential of prostate cancer. So I stepped in the hit identification phase of this drug design process. It is not the very first steps, let's say, but is at the beginning enough. Before the hit identification, there is the what you can call fragment screening or similar, where you point out even smaller molecules than those depicted here in the slide that in jargon are called warheads. So they are functional group that bind in the depth of your putative target site, and that are those that you then look for in the other steps of this process. So you start, of course, with a virtual screening of libraries where you screen hundreds of thousands of compounds containing the warhead I was speaking of. Then, after scoring the molecules, you test in vitro if they actually work on your protein. Try to get the crystal, analyze all the crystal of the complex to understand which are the interactions that are best performing, which interaction you don't want to be in your molecule. You do this cycle a few times until you reach molecules of a certain complexity, and you cannot go in this way anymore. So you can't screen just libraries, but you have to start planning different strategies to improve the performance of your molecule. The phase is called hit optimization or hit to lead, it depends on. And there are, of course, also in this case, different strategies. What we usually were doing is approaching either with fragment growing or fragment joining. Fragment growing, I believe, is what you can more easily imagine, is just expanding progressively the hit you found in the previous campaign. Fragment joining is a bit less let's say straightforward, but it's just as the name says, combining two or more fragments into one more potent compound. So what we did, starting with the fragment growing was taking one of the best hits we had and analyzing the structure both of the compound for the chemistry related part and synthesis related part, and of course the interaction with the protein, and point out which portion of the molecule could be expanded and how. And so you start and design libraries of compounds that where you take into account one position at a time. So there is one library with R1 modification, one library with R2, one with R3. You dock and computationally select which are those that has the best hope to go on with the process. One thing I omitted is you have to be able to synthesize the molecule. We had to lean on external company doing the synthesis, but it happens also with the best group of collaborators you might have. You may design an amazing synthesis path on paper, and then it can just fail, and you don't know why. So there is a big if before having the molecules to test in the lab. We do, as usual, binding assay, co-crystallization, analyze the structure, and then we put together the data to understand which modification are the best performing and why. As you can see here, we saw that the best position to modify was the R2, because with the best performing substitution, we had an 8-fold increase in the potency, followed by R1. What we actually thought it was the better, so the R3, it is also the most straightforward from the chemical point of view, because you can do amine coupling and generate a huge amount of analogous, was the one that was less impacting. At this point, what is left to be done is reductive, but it's okay, is to just repeat the previous project, the previous cycle, so designing libraries and then scoring, synthesizing, and testing, but this time putting together the best performing modification in order to have these small libraries of compounds that should be, let's say, the superheroes of these series, and hopefully a few of them would be so good that can be moved on with the development pipeline, and so go to in-cell test and on and on. Fragment joining, as I said before, is of course joining together different molecules. From the practical point of view, is a little bit more easy easier on the first part. You start from the co-crystal of your head, let's call it like this, and then you soak it with additional fragments that usually, even in this time, are screened for their capability to occupy the space that is not already occupied by your head group. And then you analyze again the structure and try to design libraries, and again having them synthesized, of molecules that are the combination of the two fragments you have seen in the co-crystal structure of the ternary complex. So the pipeline goes on also in this case with binding, co-crystallization, even because the two approaches are not mutually exclusive. So you can start doing fragment joining and then explore your new family of ligand and see in which way you can punctually modify them in order to go and achieve better potency, better selectivity, and so on. As you can see here in the top right, in this case, we had quite a nice result because we had an increase of 14 times of the potency of the inhibitor. A little disclaimer, the number you see, speaking of inhibition in absolute terms, are not so good because usually you are, the most common system are enzyme inhibition, and you can reach order of magnitudes better inhibitor going to nano or even picomolar affinity. Here we are inhibiting protein-protein interaction. So it is already per se way weaker than the interaction between an enzyme and its substrate. And also, when you are speaking of protein-protein or protein-DNA interaction, the relative concentration of the species is way lower. And so you compete both with the affinity of your molecule, but also with the concentration of that your molecule can reach in the system you are trying to tweak. So this is the end of my presentation. I hope to at least to have been able to give you at least an idea on what is going on in the lab, experimentally speaking, and especially of what we can do in the lab using the instrument of structural biology, because you have seen at the beginning, the two groups are investigating extremely different topics, but a lot of the technique, well, almost all the techniques I showed you in this presentation can be applied to more or less, but it is a big more or less, but for a lot of different proteins, and they are in different labs in the world. So the moment of the thank you in premise to the lab that welcomed me and to whom I'm collaborating day by day, and we are starting to getting results. So next time I hope to show you some very nice story coming directly from the lab instead of just beautiful pictures. And then I have to thank the group of my previous lab in which I did the work I presented in the second part of my presentation, and the collaborator from the University of Zurich that did all the computational part for that work, and of course you for the kind attention.